Not like they would in the first place. Ready?
Yeah.
All right. All right, hello everyone, and thank you for coming to the dissertation defense of Amanda Sharp. I'm super excited for her to tell you all about the work and the different projects she's had for the past few years in the lab. Amanda came to Virginia Tech in 2016 by way of Chico, California and Charlottesville, Virginia. She completed her bachelor's degree in the biochemistry department here at Virginia Tech and had an ASBMB distinction with that degree. We were lucky enough to keep her for her master's in life sciences, and she completed that project studying the druggability of kinases. Learning to want to code more and get more into the data side of things, she joined the GBCB program in 2020, shout out GBCB, and has been working on her PhD since then on drugging difficult to target interfaces in membrane proteins. Along the way, Amanda has worked on a number of projects, including the spike COVID protein, doing different free energy calculations and getting a publication in Cell, while also working on GP140, an envelope virus protein that we have in our lab, and having a publication in the Journal for the American Chemical Society. But today is all of her work on different proteins that we need to learn to target more and more effectively. And I'm super excited to have you all be here to support Amanda and to hear about her work. Let's give her a round of applause to start this.
Hi, everyone. Thank you guys so much for coming across campus today. Thankfully, it's a nice day, but I know that most of us work on the other side. So really appreciate your time today to be here, and pretty decent turnout, so I really appreciate it. So today I'm going to talk about some of the projects I've worked on over the last couple of years during my graduate studies. That'll be refining computer-aided drug design routes for probing difficult protein targets and interfaces. So I'm specifically interested in how computational techniques can be applied to cancer drug development. Cancer is the second leading cause of death in the United States, just behind heart disease. It results in over 600,000 deaths and 1.8 million new cases of cancer each year in the United States, making it very prominent and a drive to identify new novel targets for drug discovery purposes. So cancer typically follows a four-step or four-stage process for development. Stage one is the initial site, and it's early stage cancer, so smaller site in the tissue. Stage two is larger in size and has the potential to infiltrate the lymph nodes. Stage three is very similar to stage two, where it's larger in size and at this point has infiltrated the lymph nodes. And then stage four, which is also called advanced cancer or metastatic cancer, is where that cancer has been spread to other tissue types. So I'm specifically interested in lung and brain cancer because of their low 5-year survival rates. Lung cancer also just because of how prominent it is. I think it's the third most common type of cancer of all cancer types. And then specifically glioblastoma multiforme because it is one of the lowest 5-year survival rates for cancer types altogether. So we utilize computational research strategies as an advantageous avenue for studying difficult to drug proteins involved in cancer. So when I'm referring to difficult to drug protein interfaces, these are typically proteins that are difficult to explore experimentally or have been deemed undruggable in the past. So I'm talking about membrane-bound proteins and protein-protein interfaces. So some experimental workflows for exploring these structures, highly focused on structure-based methods, but with membrane-bound proteins specifically, that can be really difficult just because of the environment that membrane-bound proteins require, and a lot of times crystallography and cryo-EM techniques requires that protein to be in a solution. Although there have been a lot of advancements in the recent years using cryo-EM and exploring membrane-bound proteins. Mutagenesis is also another approach for exploring drug proteins, where you mutate specific residues that you anticipate to be involved in either activation, complex formation, and so on. And then a more recent advancement in exploring protein-protein interfaces is the protein painting technique that our collaborators at GMU have identified, utilizing molecular dyes and mass spectrometry to identify hotspots at these protein-protein interfaces. Now, all of these research techniques are really great for exploring these, but we're proposing computational techniques to alleviate some of the cost of doing these different techniques or accelerating the overall workflow. Additionally, with protein-protein interfaces, there's no specific pocket that you can target, which makes it even more complex for drugging. So you can't just like fit a small molecule in there easily. You have to really get creative with targeting that interface. And both protein-protein interfaces and membrane-bound proteins are highly complex and sample multiple conformational states. And that's important to understand as well. So today I'm going to be talking about 3 different projects. I'm going to talk about PD-1 and its application in lung cancer, OR2T7 and glioblastoma multiforme, and SPNS2 and metastatic cancer. So all of our research techniques that for all of my projects, at least today, utilize molecular dynamics simulations or MD simulations. And essentially MD simulations can be described by this one quote that everything that living things do can be understood in the terms of the jigglings and wigglings of atoms. So essentially what MD simulations allow is for dynamic movement of atoms in space. And this is based off of the analytical force model, force equals mass times acceleration, or Newton's second law. So a little bit of math. I won't go too much into the math, but with Newton's second law is the basis of our force fields. And we also know that force is equal to the negative derivative of potential energy over the derivative of position. Now we always know position. Our input structures have 3-dimensional coordinates of every atom in space, and we can solve for the potential energy. So our potential energy is the summation of our bonded and non-bonded energy terms. Breaking that down even further, we have our bond stretching, bond angles, dihedral angles or rotations. And those are our bonded terms, and our non-bonded terms are our van der Waal, electrostatic interactions, and everything outside of that. So these are the parameters that we're able to define how our atoms can behave and the movement in our simulations. So now you have a little bit of understanding of what we do in our lab, or at least the premise of all of our work. I'm going to go into my first topic, which is discerning agonism and antagonism for disrupting the protein-protein interface of PD-1 and PD-L1. So PD-1, or the protein cell death protein 1, is an immune checkpoint modulator protein that's expressed on T cells. So PD-1 has been identified to play an important role in many different cancer types, but has been heavily explored in non-small cell lung cancer, or NSCLC. So essentially what happens here is that PD-1 expressed on T cells, and then cancer cells can express PD-L1. I know it can get really confusing here, so I'm sorry about that. The naming, not my idea. But it can express its native ligand, PD-L1, and the interaction between PD-1 and PD-L1 allows for cell proliferation. So it allows cancer cells to evade the body's innate immune responses. So the proposal is blocking the PD-1, PD-L1 interaction using some types of drug or biologic to force cells into tumor suppression pathways. Now, this is a well-studied pathway generally. There are emerging therapeutic strategies. In 2020 was the release of Keytruda. Keytruda does block the PD-1, PD-L1 interaction, targeting PD-1. So there are a lot of immunotherapies now currently on the market targeting this. So immunotherapies are great, and they're highly specific for the target, but they do have poor pharmacokinetic profiles and can circulate in the body for days, weeks, or months. So that lengthening of time that these immunotherapies are in your body can be really hard with treatment when you're already having chemo. So there are no chemotherapies available for targeting this interface. There is no clear binding pocket, which makes it exceedingly difficult to develop a chemotherapy for. But there are benefits to chemotherapies because of their smaller size. They have better pharmacokinetic profiles and can be excreted a lot faster than the immunotherapies. But because of their small size, they have their intrinsic ability to have off-target hits. So we propose utilizing peptide therapeutics to bridge the gap of benefits between immunotherapies and chemotherapies. So an improved pharmacokinetic profile with small size of the peptide, but improved specificity for our target protein we're interested in. So with our collaborators at GMU, we have developed a peptide construct. So essentially they were able to identify the first designer peptide by using an alpha screening. So that is what this is here. So they're measuring the percent activity, which is the activity is complex formation. So the interaction between PD1 and PDL1 interaction, that's what they are calling their activity here. And they were able to identify that this MN1.1 peptide was able to block PD1, PDL1 interaction at micromolar concentrations. So what this peptide was constructed with was essentially just residues 112 through 128 of PDL1, which you can see highlighted in purple here. So it's literally just the beta sheet that is at the interface between PD1 and PDL1. So they just cleaved that section there, and it ended up actually working pretty well as a starting point for peptide development. So just want to get you some background of when I say MN 1.1, that is what I'm talking about here. So I'm going to skip forward a little bit. We've worked with our collaborators quite a bit. We only have a certain amount of time today. I'm sorry, guys. We worked with our collaborators quite a bit to identify a very potent peptide inhibitor or peptide inhibitor of PD1, PDL1 blocking. That's MN 1.4 is what we've called it. But unfortunately, when analyzing cytokine levels and T cell activation, we're observing that this peptide behaves similarly biologically to PDL1. So what this means is that our peptide is still activating the structure the same way that PDL1 is, creating an environment for cancer cells to continue to grow. So that's not necessarily what we were wanting. So we were wanting to develop an antagonist, which would inhibit the activity of PD1, and we accidentally developed an agonist. It can be really complicated when you're doing drug development. You know, your first initial plan is just let me get that to bind really well to that interface, and then it goes haywire and does the opposite of what you're anticipating. So that's what we were observing here. So I kind of had to pivot a little bit in my research studies because I'm still interested in the application of this peptide development in cancer. So that's what I really had to do. I was interested in looking at the differences between agonism and antagonism in PD1. So the research goals for this project were, can we identify PD1 activation mechanism? Are there structural changes that can help us predict an active versus an inactive state in PD1? And what features can we exploit for the development of a PD1 targeted cancer therapy? So it gets a little complicated here. So I'm trying to give you guys as much information here to follow along with this. Again, with PD1, PDL1, it gets really confusing. So we have our inactive systems, which is PD1 apo, which means nothing's bound. And we have our PD1 null peptide. So this null peptide is actually constructed computationally with us and experimentally validated to not block PD1, PDL1 activity. And then we have our active structures, PD1 in complex with PDL1 and PD1 in complex with MN 1.1. So I also want to emphasize here that this research project, I'm only interested in PD1. I think like further down the line, I'll be interested in the interactions between these different biologics, but for here, I'm really looking at what is being elicited with that protein structure in response to agonistic binding or antagonistic binding. So the first analysis I did, I looked at residue distances through our simulation for every residue in the protein structure. So that is depicted here. I did a statistical test to analyze what were significant residues in the differences in distances there. So we were able to identify 7 different domains that had residues that were significantly different in their distances to other residues. So that's just depicted here. I also wanted to do further analysis on this. So I generated a distance matrix and analyzed the differences between the distance matrix between PD1 apo and PD1 in complex with PDL1. So that's depicted right here. We see a few different regions that are of interest, which I circled here to be easily referenced to. And what we see in C, this is referenced right here, was really interesting because that region seems to be highly stable in PD1 and actually really highlighted the difficulty of studying this protein structure. PD1 is extremely stable, and it has this disulfide bond right here that is important in the structural stability and integrity of the structure. They've done experimental work analyzing this disulfide bond here, and essentially PD1 unfolds if this isn't there. So it's very important there. I also highlighted that there's possibly a pi stacking interaction helping with structural stability here as well. And while there were differences here, there's nothing really at this point that I've identified that was difference in this region, but really highlighting how stable this structure is overall. In D, we see D4 here, and a loop in the back of the protein structure, which is not highlighted as a significant region, but D4 was identified to have significant differences in distances. So that was interesting there. In E, we're seeing D4 highlighted again with its distance to D5 here. And that was very interesting to me because D5 is the neighbor of the membrane proximal region. So D7 is the membrane proximal region of the structure. So and then F, that's where we see differences with that membrane proximal region of D7. So essentially membrane proximal, I mean that we have this structure of PD1, which it is an incomplete crystal structure. We anticipate that following that D7 region is the membrane embedded region. So highlighting differences in the membrane proximal region can give us a little bit of insight on how agonist binding is eliciting activation responses in this structure. But I also think that D4 region seems to be very important based off of our analysis here as well. I also highlighted this D3 region here, which is this big yellow loop region, which I'm going to kind of discuss a little bit more later. So the next thing that I did was analyze the secondary structure of our protein. So these are our cluster structures of all of our different systems. So I do have it labeled kind of small here, but this is APO, PD1 with the null peptide, PD1 with PDL1, and PD1 with MN1.1. So the top two are inactive structures, our bottom two are active structures. When analyzing the secondary structure of the full protein, we're honestly not seeing very much difference. Yet again, going back into how stable this structure really is. But when we look at that D3 loop, which is right here, we're starting to see some secondary structure differences. So I also highlighted it here, so it's very abundant that there's differences there. So I analyzed the secondary structure there, and we were able to see that there are differences in beta sheet formation versus coil formation there. The beta sheet formation in PD1, PDL1 is very stable, where it is not as stable in our PD1 APO structure. So that was something that was really interesting and can be important to explore later. Looking at the different systems with the peptides, there were a bit of inconsistencies there. So for that purpose, I did continue most of my research focusing on PD1 APO and PD1 in complex with PDL1. There might be some different inhibitory mechanisms with those ones, or binding mechanisms, not inhibitory. So the next thing I did was calculate per energy residue calculations throughout our protein structure. And these ones are highlighted as the most important or have the biggest difference from our APO structure. And immediately I see 75 and 76 as very different and much lower energy in our active states versus our inactive states. So looking at where those positions are located, that is in that D3 region as well. And you can visually see what's occurring here. So our green is our null and our red is our APO. These are more outwardly facing compared to our MN1.1 and PDL1 complex structures. So it appears that that stabilization of that beta sheet is bringing those residues further down and decreasing the energy of those residues, having some impact in activating the structure overall. And the last analysis technique that I used for this project was doing principal component analysis to explore the extreme positions and phase space occupancy of our simulations. And what we highlight here in our PCA, it seems that there's a lot of overlap, but that is actually really normal for PCA analysis. They are the exact same proteins. We anticipate them having mostly overlap there, but we do see differences overall. So in our PCA, we're also able to extract the most extreme positions that our atoms have in our simulation, and that is depicted in the noodle looking like figures at the bottom. So the red, again, is our APO structure. The deep purple is our PDL1 structure in complex with PDL1 structure. And we're immediately seeing some differences in variation in our D3 loop. Seems to be a lot more movement in our D3 loop and some difference in the way that the movement is occurring in that D4 loop. We're also highlighting a few other loop regions that seem to have variation in movement there. But I also wanted to quantify this data. So I analyzed the differences between the C alpha carbons in each extreme position to see how much that movement is occurring in each system. And what we're able to highlight here is that the majority of the difference in the movement is in this D3 region here. Again, that region that we've repeatedly highlighted as an important region in agonism versus antagonism, and it appears to be fluctuating a lot more in our apo or inactive structure and is more stabilized in the active structure. So it appears that the beta sheet formation in that region is stabilizing the fluctuation in that residue. We also see differences in the L3, loop 3, which also is connected to that D7 region, further giving us a way to describe the activation mechanism in PD1 overall. So collectively, what our simulations are able to describe, essentially we're given a baseline for analyzing agonism and antagonism in further MD simulations. Our simulations demonstrate that D3, D4, and D7 regions appear to be important and an indicator of ligand binding, or if a ligand is an agonist versus an antagonist. Appears that the stabilization of that beta sheet in the D3 region is eliciting the responses of agonism. And if you're wanting to develop a drug that is targeting PD1 for cancer, you should avoid targeting residue 75 and 76 to avoid hyperstabilizing that beta sheet formation there. But if you want to take a different approach for autoimmune diseases, you would definitely want to target that residue. So there's other applications for that as well. So there's some future directions that I would like to see done with this project going forward. We did a computational alanine scan of the peptide, so mutating every individual residue to an alanine and to identify specific residues that are important in eliciting those responses. And I would like to be able to construct a machine learning model for prediction of that. I think it would be interesting to model in the membrane embedded region of PD1. I anticipate that's just an alpha helix group that is embedded in the protein, and do MD simulations in a membrane environment. I anticipate some of those loop regions that were fluctuating may have some membrane interactions. So it'd be interesting to see that as well. Okay. Take a quick sip of water. So the next project I'm going to discuss is the project for OR2T7, a novel prognostic marker in glioblastoma. So glioblastoma is a really dangerous brain cancer type where it has less than 5% 5-year survival rate, emphasizing how important it is to develop novel therapeutic options for targeting this. So our collaborators, Dr. Ananda Krishnan, who's a member of my committee here and present here today, did some bioinformatic analysis on GBM samples, and they identified this mutation D125V that is present in glioblastoma patients, but not the general population. Experimental works were showing that this mutation downregulates RHO-B, P3, Cas14, or all these different genes here, all genes that are important in apoptotic pathways. So we wanted to use our computational research strategies to explore why this is happening here. So this is a homology model of our OR2T7 structure here, and describing a few of the different helices of interest in the protein overall. I also have this highlighted ICL2 or intracellular loop 2 region here, which is where that mutation is occurring. And essentially, we hypothesize that there are structural determinants that influences G alpha subunit binding that is influencing the differences in expression of these apoptotic genes happening. So to orient you in a few different helical groups that I'm going to be talking a bit about today, we have our TM5 transmembrane domain 5, 6, and 7. These regions have been identified to be important in G alpha subunit binding. So we have this depicted in experimental data here. So this is adenosine A2A, which has been a crystal structure of these different structures. And this is a closed conformation. So in TM6, in a closed conformation, will be closer to TM7. In an open conformation, TM6 will be further away and bend away from that TM7 region. So I'm really interested in seeing in my MD simulations if I'm able to observe this phenomenon. And TM5 has essentially been identified to be a stabilizing feature of OR2T7. There we go. I don't know why I took a hiccup there. Additionally, there's a few other regions I'll be discussing, the ICL2, which I highlighted before. That is the location of our 125 mutation, and ICL3, which is between TM5 and TM6. So the research goals and questions for this project are: what are the global structural impacts of this mutation? If we can identify local impacts of this mutation, and does this mutation have the potential to obstruct G-alpha subunit binding, thus downregulating these different genes that were experimentally determined? So the first thing I wanted to highlight in this project is the dominant morphology. So we're able to determine dominant morphology using clustering, and the largest abundant cluster is our dominant structure. And immediately I'm noticing some differences in these structures. We see it appears to be some differences in compactness overall. Here's our wild type structure in the teal, our mutant structure in these tan colors here. It seems that the wild type is less compact compared to our mutant structure. So that's something I wanted to further explore as well. Also, the TM6 region appears quite different in positioning in the two systems. It's kind of hard to see here, but we have this region right here, that's TM6. This is TM6 here. It's different between the wild type and our mutant structure. So that's something I wanted to further explore. So it appears that the mutant structure is having less variability in its structure, where our wild type appears so far to have a lot more flexibility. So I wanted to further analyze the compactness hypothesis there. And oh man, that does not show up very well on these screens. I'm sorry. What we're seeing here is that we use radius of gyration to be able to analyze compactness of the system. And the mean of our compactness is very similar between our structures. But when you look at the variation between our structures, that's where we're seeing a lot of differences. So it appears that there is difference in compactness that I was observing from our cluster structure. So you can kind of see it here, but it's real. The standard deviation there is much closer to that mean. So I wanted to see if that compactness was having global impacts of the structure. So we did our residue-residue interaction distance like we saw in the PD1 project. And essentially we're seeing a lot of changes happening here. So the red and blue is highlighting the differences between our simulations. So lots of differences here. Also wanted to highlight this 125 region, which is the location of our mutation. It also appears to be closer to that ICL3 loop in our simulations, and that is between TM5 and TM6. I also wanted to analyze the positioning of our protein in a membrane. So I may have not mentioned this earlier, but our protein is simulated in a membrane environment. I wanted to see if that could be impacting the variation and flexibility of our structure. And we are observing that our wild type structure is higher in the, our wild type structure is lower in the membrane, where our mutant structure is sitting higher in the membrane. So that alone has the potential to obstruct the overall flexibility in the different helical groups. So the next thing I wanted to do was look at what is locally occurring, what in that 125 region is occurring that causing these different changes. And we did secondary structure analysis. And what we're seeing is that in our wild type structure, this region between that loop region is actually stabilized with an alpha helix group. And our valine in that 125 position seems to destabilize that alpha helical formation. What's really interesting there is that valine and aspartate have similar propensities to be present in an alpha helice, but they're behaving quite different in our simulations here. So it appears that maybe because it's near the membrane environment that it is destabilizing that helix formation, but this is likely what is influencing the differences that we're observing. So the next thing we did was PCA on the structure. We also did PCA on the full structure, and basically they were identical. They overlaid perfectly, which again, they're the same protein, this one just with one mutation. That's what we expect to see in our simulations. But when we analyze the principal component or do principal component analysis on the TM5, TM6, and TM7 regions, those regions that are important in G-alpha subunit binding, that's where we're starting to see some differences. So we can see that our wild type structure is sampling more space on eigenvector 2, where our mutant structure is sampling more space in eigenvector 1. So we can extract our extreme positions like we did before in PD1, And we're definitely seeing movement differences here. So we're seeing more of a scissor-like movement in our wild type structure and more helical flexibility there. And it appears that our mutant structure is showing more helical rigidity and less flexibility overall in that structure. So another analysis I wanted to do is look more in depth about these TM5, TM6, and TM7 regions. So I looked at the angle between TM6 and TM7. So I wanted to see if this angle was changing in these structures. And what we observed is that this angle is greater in our wild type structure on average. But interesting is when we look at the probabilities of the positioning of this angle. So both of the simulations show a trimodal distribution of angles, where we have our open, which would be the largest angle, our intermediate, and our closed conformation. So these have been observed experimentally as well. But interesting is that our mutant structure appears to sample mostly a closed conformation, and that its open conformation appears to be more similar to what an intermediate in the wild type structure would be. So that definitely has the potential to obstruct G alpha subunit recruitment or binding in general. So we can compare this to experimental work. So we have those adenosine A2A crystal structures that we analyzed before. And essentially, analyzing the angles in that structure, we were able to observe that only our wild type structure was able to get an angle similar to the experimental work. So our mutant structure was not able to obtain an angle large enough to be able to accommodate a G alpha subunit collectively. So this is definitely promising for being able to describe how this mutation impacts the overall activity or the downregulation of those apoptotic genes. So collectively, what we're able to describe in our MD simulations with this project is that valine 125 destabilizes the secondary structure in that ICL2 loop. And that has shown to impact the TM5, TM6, and TM7 domains. It appears that there are changes between the open and closed conformation, which we know experimentally can impact G alpha subunit binding. And essentially, collectively, what we're showing, though, is that our wild type is sampling or is showing more helical flexibility overall, where our mutant structure is much more helically rigid. So I also have a depiction here to kind of describe how this is impacting G alpha subunit binding. So the gray is a G alpha protein. And you can see this is an open conformation from our simulations of the wild type structure. You can see that it can fit in there quite well. But when you have our closed conformation, it is obstructing that region overall. You can see that it really can't bind here at all. So this also provides an avenue for those who are wanting to explore GPCRs and analyze single site mutational impact and to use for pharmaceutical drug discovery purposes. Future directions for this project, I think that there are a few different avenues I would be really interested in looking at. I think virtual screening of a large database of different ligands would be really important to understand activating what molecules can activate OR2T7. I think it would also be interesting to extract multiple conformations of our GPCR we were observing in our simulations in doc, because the extracellular region of OR2T7 is very is random coils, so it can move a lot. So there's going to be multiple ways that a ligand can bind there. So being able to sample multiple states, multiple places that maybe it has multiple different pocket sites, I think that would be really interesting to do on this project. I think enhanced sampling methods would also be able to further analyze our conclusions that we're making in this analysis. And then I think MD with a G protein using steered MD and umbrella sampling can help us understand the energy of the G protein release, further supporting the conclusions we made here. Take another sip. All right, so the last topic I'm going to discuss today is a sphingolipid transport protein, spinster homolog 2. So spinster homolog 2, or SPNS2, is the main transport protein that transports sphingosine 1-phosphate, or S1P, from the extracellular region into the, or to the cytosol, from the cytosol to the extracellular region, sorry. So S1P has been identified to be elevated in numerous different disease states, but has been specifically highlighted in metastatic cancer. Essentially, S1P provides the right microenvironment to support metastatic cancer, can stimulate angiogenesis, inflammation, and cell migration, basically providing all the avenues necessary for a cell to pick up and go to another tissue type. So SPNS2 has been that emerging target of interest of researchers because it has been, it is the main transport protein that exports S1P out of the cell. And some research has shown that this R119A mutation significantly decreases extracellular concentrations of S1P. So we do work on this project with some collaborators in the chemistry department, and they have identified some really awesome inhibitors for binding SPNS to inhibit S1P translocation. But we don't really know how it binds at this point, and it's really important to understand the structural mechanism to further enhance the drug discovery process. So we have a few simulations that we did with this project. We created a homology model of SPNS2, and we simulated it in a membrane environment. We have our MD simulations, I should just say just MD simulations of our APO structure. So we have 4 replicates each of those. We have our SPNS wild type APO and SPNS mutant APO. We also did a few MD simulations of S1P in the environment to see if we can observe translocation, and we did 6 replicates of those each as well. So the research goals for this project, I wanted to see if we can identify the transport model for SPNS2. I wanted to see if we can identify how that mutation impacts the structure of S1P, thus leading to the inhibition of transport, and if we can identify any residues that can be exploited for drug discovery purposes. So when I talk about the transport models, this is kind of what I am interested in investigating. So SPNS2 is a major facilitary superfamily protein, and they can follow 3 different solute transport models. I have depicted the rocker switch model, which essentially it goes inward facing like this. And when a ligand is bound, it is being transported, it goes to an outward facing V like this. They both move simultaneously together. There's also a rocker bundle where only one bundle will move and one stays like at a 90-degree angle. And then there's an elevator model, which is a lot more complicated, but it does like some weird elevator type movement like that. So I'm interested in seeing what type of solute transport model is occurring there. Additionally, it follows a secondary active transport mechanism, which means it doesn't get transported by itself. Something else is there to help move it through. So here I have depicted an antiport transport model where a molecule will come in from opposite side of the protein, where the S1P would come from this side, or symport where they get transported together. So those are things that I'm interested in looking at in this project. So looking at my cluster structures here, I immediately see some interesting things where our simulations have ions present in the simulation. So that was something I got really excited about. We're seeing cations in there, possibly observing some secondary active transport. So these are our wild type systems. And our mutant systems, we're seeing some interesting things as well, where we're seeing multiple cations in our simulations. It's kind of hard to see with the orange and the red, but I promise you there are multiple ions in those as well. And replicate 4 somehow got a lipid stuck in there, but sometimes those things happen in our simulations, some weird things happen. So the first thing I wanted to explore is if we can identify the rocker bundle, rocker switch transport model in our simulation. So I did so first by analyzing the angle between the helical bundles. So we have our N-terminal bundle and our C-terminal bundle. So I analyzed the angle between those. And what we're able to observe is that the mean angle between our wild type and our mutant, the mean is smaller in our wild type structure, but has more variation overall. And our mutant structure has a larger angle between those, but is much more rigid and stable. I also analyzed each individual bundle relative to the membrane normal. Essentially what that means is just relative to the center of the membrane. I wanted to see how those helical groups sit in the membrane. And what we're observing is that those angles in our wild type structure are pretty much identical to each other. So they're sitting similar to each other rather than one being more skewed over to demonstrate that rocker bundle. So this is really highlighting, and this is likely a rocker switch model, we're seeing it pretty similar in our mutant structure as well. It's a little bit skewed based off of the scale of my graph here, but it's only about 4 degrees different. So that's not really that big of a difference there. But collectively, this is showing that first, that the wild type structure is has much more helical flexibility, where the wild type is more, or the mutant structure has more rigidity in its helical bundles, and that this likely follows a rocker switch transport model. So I wanted to understand how the rigidity is impacting the overall structure. So major facilitary superfamily of proteins have these two different distinct and important helical groups. The gating helices, which are involved in interdomain conformational shifts during translocation, and our cavity helices, which are what it sounds like, those are the ones where the binding cavity is, or where the ligand will bind to the structure. And we are seeing some differences in our simulations here. So I have it depicted in what the gating helices are here and what the cavity helices are here. We did PCA on this, and we're seeing some interesting differences between those. We're seeing 2 groups because we have 2 groups of helices, so they're not going to be necessarily overlapping on each other. But when we look at the extreme positions here, we see, again, more helical flexibility in the wild type structure. You can see in the H11, helix 11 here, more movement here. Oh man, this is blocking now. Move me over here for a second. More helical flexibility in 11 compared to the mutant structure, and more flexibility and movement in helix 8 and helix 5 in our wild type structure compared to what we're observing in our mutant structure. So I'm going to move me back. Hold on, sorry. Oh, I'm stuck there now. Okay, I'm stuck there now. I'm sorry. And then in the cavity helices, we're also seeing a lot more helical flexibility in those, where we see that there's just general fluctuation between positioning in the cavity compared to our mutant structure. So this is further supporting that our wild type structure samples more flexibility in its helical domains, where our mutant structure is much more rigid, which definitely has an ability to impact substrate binding. So maybe that the flexibility can support translocation or support the interaction with the channel environment. So that was something that was really interesting. So I'm not sure if anyone else has experienced while you've been doing your research, where another lab publishes something very similar to what you're looking at. Unfortunately, that did happen to me while working on this project, and I try to take it with positive notes here, but another lab published something in Nature exploring these different topics in SPNS2, but in can't pronounce this, Piphomonas neptunium, so it's a bacterial strain. So it's not exactly what we have here, but it is a crystal structure of that homolog, where ours is a homology model. But in their work, what they were able to identify is that glutamate 207 protonates and liberates arginine 119, allowing it to bind to a substrate and trigger local and possible global conformational changes. And then in contrast, only when aspartate 118 is deprotonated and interacts with arginine 200, it can glutamate 207 function as a intracellular master switch. So when looking at this immediately rings a few bells in my head. These are all charged residues that they're identifying to be really important here. So I'm wondering if there is a salt bridge network occurring in this protein structure and supporting the translocation in that capacity. And that needs to be moved. Oh, there we go. Okay, no, I'm moving. All right, I'll minimize it. People on YouTube don't need to see my face today. So I wanted to further analyze the role of salt bridge formation in our simulation. So I actually analyzed every possible charged interaction in the channel to see if there's any interesting salt bridge interactions occurring. And what we're able to observe is that there are differences, and especially some of the highlighted from this paper, differences in our mutant and our wild type, wild type and our mutant structures here. So what we see at first with 118 and 200, there are differences in distances there, whereas the wild type structure has its distance between 118 and 200 close enough to be considered like a salt bridge or form a salt bridge there. Whereas our mutant structure is not close enough for that. So these are in the nanomolar ranges. We'd want to see it at 2.5 angstroms. We are not observing that distance there. 119 and 200, we're definitely seeing some possible salt bridge interactions occurring in between those two residues as well that we're not observing in our mutant structure. And at first that really didn't throw a red flag for me because an arginine to an alanine is really big change, and arginine is bulky and much longer, and that could explain the difference in distances there. But the fact that we're also observing it in other residue or other salt bridge network residues as well was really interesting to me. And we also identified another possible salt bridge forming interaction between 119 and residue 472. We see that it's not super consistent in its distance to be able to form a salt bridge network, but it could be important in the translocation process overall. So I also have those interactions highlighted here. These are wild type structures. You can see how close they are in proximity to each other. But what's happening is that this R119A mutation is actually impacting the salt bridge network, thus reducing the transition between an inward facing conformation to an outward facing conformation. So I also wanted to see if this R119 mutation impacts the electrochemical gradient within the channel. So we're going from a charged residue to a non-charged residue, a non-bulky residue. So I wanted to see if that is impacting the electrochemical gradient. And what we're observing is that, so this is analyzing the distance of potassium to the membrane normal. And this is really analyzing like how frequent, man, I really picked the wrong colors here, guys. I'm so sorry. So, I mean, you see a little bit here, which it is light, and it means it's not like really stable in that same region. We have all replicates stacked on top of each other. So when you see something in the pink region, it's common in all replicates. But essentially what we're seeing is that potassium does get recruited into the channel, but it doesn't stabilize in any specific region. It is fluctuating where it is. Whereas in our mutant structure, once a potassium gets into the channel, it likes to stay near that salt bridge residue or those salt bridge networks there. So this is really supporting that there is a difference or a change in the electrochemical gradient, which is the mutant structure requires additional cations to stabilize those interactions. We also notice that there's potassium or sodium ions also entering that channel as well. So we also did our simulations with S1P in the environment to see if we can identify how transport is happening, how S1P is recruited into the channel of SPNS2. Unfortunately, only 2 of my 6 replicates had any interactions with S1P. And that's sometimes what happens when you take a chance to see if we can randomly have it go into the channel. Interestingly, those 2 replicates are also binding different from each other, but we see some similarities with the initialization of that interaction. So these are the interactions with all the interacting residues. So sorry, it's kind of hard to see here, but we have some similarities in this 300 region that is similar with our replicate 5. So it appears that those residues are likely most important in the recruitment of S1P interacting with SPNS2. Now with our replicate 2, which is here, it shifts in its interactions over here and becomes highly stable. Essentially in that replicate, what happens is that S1P just gets sandwiched between the protein and the membrane. But with the consistency of these interactions with the 300 range, it is able to enter the channel and kind of sit there. We didn't see translocation really happening though. So I also did fingerprinting, time series fingerprinting to identify residues that were important in binding or in interacting with S1P in our simulations. We're able to further highlight the shared residues of the 300 region in our recruitment, but it appears that recruitment into the channel is can be highlighted with the residues 3-enene 216, arginine 227, and methionine 230. So those are residues I think would be very interesting to further explore and potentially residues that can be important in exploitation for drug discovery purposes. So MD takeaways, what we're able to observe is that this mutation, the R119A mutation, impacts the electrochemical gradient of the SPNS2 channel. By doing so, it alters the salt bridge network, which we know to be important in substrate translocation. Collectively, it's able to impact the helical plasticity and the gating and cavity helices, and that's how it's able to impact substrate translocation overall. We were able to identify some very preliminary information with our simulations of the channel residues of threonine 216, arginine 227, and methionine 230. And I think these can be exploited for drug discovery purposes. And very preliminary, again, only 2 replicates demonstrated any binding in our simulations. But with the fact that our S1P was able to both enter the channel and get sandwiched between the protein and the membrane, we think it's possible that this protein is a flippase where it actually recruits S1P from the membrane, flipping it into the channel and then exporting it in that manner. But we need to do a lot more future directions with that to make any conclusive statements there. So future directions, I think it would be interesting for the MD simulations to do MD simulations with altering protonation of all the salt bridge residues to see if that could help elicit a conformational shift or simulate with proton in the solution. That one Nature paper that came out, unfortunately very close to me finishing my work, they did protonation analysis to see if that's what's impacting conformational shifts, and they did observe conformational shifts that I was unable to observe in my simulations. And got a few minutes. I'm going to quickly go through this. Sorry, guys. But we do got some exciting future information with some molecular docking results with our collaborators in the chemistry department, where we did some molecular docking. So our first thing that I did is I did molecular docking in the homology model, so the non-MD simulated structure. So that's the results here. This is our replicate 5 positioning of S1P, and then molecular docking results with the simulated structure, and we're able to better simulate S1P placement in our simulated structure. So I decided to do docking with that for inhibitor docking. I'm trying to go fast, I'm sorry, guys. And then they have a few inhibitors that they are getting ready to publish that are very potent at inhibiting S1P translocation. So SLF is their new inhibitor that they've discovered. And what we're seeing here, and this is 16D, which has been published and a potent inhibitor as well. What we're able to see is that these inhibitors are interacting with those 3 residues that I identified from my simulations to be important for exploiting for drug discovery purposes. So I was really excited to see that. I got a lot more, but I'm not going to take all the time in the world for this. I'm sorry, guys, but really exciting preliminary results in our molecular docking studies here. Also, they interact with that new residue we identified that important in salt bridge forming as well. So far with our very quick docking takeaways, it shows that inhibitors that interact with that salt bridge forming region seem to be most potent at blocking translocation of S1P. We are able to further highlight the channel recruitment residues that we identified from our MD simulations are important in inhibition. We have a lot of things we want to do with this. We want to test a lot more constructs and do virtual screening of inhibitors for this project. We also want to test some that have no functional activity for blocking S1P as well. And we also think it'd be really interesting to do docking studies, mutating those residues that I identified to be essential in recruitment overall. All right, sorry guys, this is a long day. I really want to thank my committee members today. Of course, Dr. Ann Brown, who has been a huge advisor for me since my senior year in undergrad, and I would not be here today without her. Dr. Allen for being a support since my master's degree, and my protein person that can always give me information on that stuff. Dr. Lumkool, who has been a support system in more ways than he knows, because if I need a question answered from him, I could usually just Google it on ResearchGate. But he has been an endless supply of knowledge in the MD world. Dr. Ananda Krishnan for, started our relationship with OR2T7. We're closing it out there too, and really helping me guide in the way of the data science and coding stuff, and being a huge support, just general, showing up to my seminars and everything. The Brown Experiential Learning Group, which is a massive group, but specifically I want to thank Kelsey, Aaron, and John for being basically my emotional support animals over the last 5 years. I have my undergrad students, Ethan, Alexis, and Katie, who are not here today, but they have done work on the PD1 project that has been very useful for me. I have all of my collaborators, which there's even more than this. Unfortunately, I've done a lot of projects while I've been here. The GBCB program, everyone here, Denny, of course, I again would not be here without you, Dr. Biswarp as well. You've been a huge support system for me and being able to answer random questions anytime I need them. My family, where my dad and my brother are here today, and my fiance, who have been supporting me during what feels like an endless education process, because I've been here since 2016. But thank you for putting up with this very long road that we get to conclude. So I'll take any questions in the few minutes we have.
So with your second project, it seems that the mutation is also from charged amino acid to
Hydrophobic, yeah.
to hydrophobic one, right? Have you looked into like, let's say the surface charge potential, something like that, so that it's, kind of like, because those mutation from polar to non-polar, so that in one case it stabilizes the structure and in the other case destabilizes the structure.
Yeah, well, so that mutation specifically is interesting, actually. So I haven't looked at like specifically with the charge, but what I do know is in that other OR2s, they have that valine in that position rather than the aspartate. So that was really interesting to observe. So clearly it still has some functional purpose in that area. And maybe down the line, we mutated to have this aspartic acid in that position to increase regulation of apoptotic pathways, or vice versa, all the others somehow mutated. But it still holds like functional purpose there. It's just downregulated, if that makes sense. But I haven't done any like charge specific analysis on it overall.
If that is the case, then both for your second and third project, where is that mutation is actually happened? Is that like on the outside part where the protein is actually facing the membrane, or it's actually in the inner part where it's actually kind of like?
Yeah, so the mutation in the OR2T7 project is cytospatial, and it's not actually directly involved in substrate bind or like G protein binding. That was really weird to see. I was like, okay, that's not a position I would anticipate having any impact at all. So that was really strange, honestly, at first. But with the SPNS2, that mutation is in the middle of the membrane area where the salt bridge forming. So the salt bridge forming residues are like center of the membrane. So in that channel specifically.
To take you to the disulfide.
Yes.
So the question is, do you have any proteomics data that sometimes you can trap that at a sulfidal level? In any data from proteomics? And secondly, what's the location, cellular location, that region of the protein sees? Because that's crying for regulation by thyrodoxine.
Yes. So where this disulfide bond is occurring, so D2 is between D2 and D6, between these two helical groups, or these beta sheets here. Basically everything that I've read experimentally that this does not break or form at all. This has to be there for the structural integrity of PD-1. There are some disulfide bonds that can occur in other proteins that are similar to this one, but with PD-1 specifically, this is the only disulfide bond that forms, and the break of that bond completely kills the structural integrity. It'll just unfold completely. Yeah, so it's in the very center of that, and basically just makes it really difficult to study because it's really stable because of that. Okay. Any other questions? No questions after. All right.
Thank you.
No, I'm not blocking.
I know, it's like I start for an hour and then you're like, oh, wait. Yeah, I watched it.